Well, as I mentioned in class the other day, we need to have one more lecture before the exam to finish the electron transport system. So that's what this lecture is going to be about. I want to remind you, first we talked about glycolysis, which takes place in the cytoplasm of the cell, with the end product of pyruvate, which is modified to acetyl-CoA, which goes into the Krebs cycle. And after glycolysis and after the Krebs cycle, all the carbon and oxygen that's come in as carbon dioxide, carbon, hydrogen, and oxygen in a carbohydrate like glucose, the carbon and the oxygen are gone. The hydrogens that have come in on glucose have been taken in the form of NADH and FADH2, both in glycolysis and in preparing pyruvate to enter into the Krebs cycle. And during the Krebs cycle, all these hydrogens have been picked up by hydrogen carriers, and the energy is temporarily stored there, delivered to the electron transport system. So we've concentrated on energy so far. That's the form of energy that we're talking about, is hydrogens, these hydrogens. So this is what we looked at the other day, that there's going to be 32 ATP that are produced. That's a lot of ATP compared to the other two phases of cellular respiration. Okay, this is something else that we looked at the other day, is that from glycolysis to ATP, in the Krebs cycle to ATP, you can get a total of 36 in most cells from one molecule of glucose. So there's 32 left to go. And this takes place in the membrane of the mitochondria, not inside the mitochondria, not in the matrix, not in the cytoplasm of the cell. And this mitochondria has two membranes. It's not a cell, but the theory is that they used to be bacterial cells that had a lot of potential to make energy, and they were incorporated into bigger cells, eukaryotic cells. So they've got two cell membranes that have different permeability. There's the outer membrane that's fairly permeable to different substances, and there's the inner membrane that's very impermeable. And there's also enzymes that are embedded in these membranes. These enzymes are going to shuttle electrons from one enzyme to another along this membrane. So in other words, they're going to transport electrons through this system, which is why it's called the electron transport system. So where do these electrons come from? Well, these electrons that enter into the electron transport system come from hydrogens. So those hydrogens coming from glucose. And now, these hydrogens, you probably all know hydrogen has an atomic number of 1. What that means is that it's got one electron and one proton, hydrogen molecules. And amazingly, the cells with mitochondria have the ability to split a hydrogen molecule into its atomic components, into an electron and a proton. And that's what happens when these hydrogens are delivered to the electron transport system. There's enzymes that work on the hydrogens and break them into an electron and a proton. The electrons are transported along this system of proteins, enzymes, so it's the electron transport system. The protons, which in all of these figures, the protons are going to be designated by an H+. That's not just hydrogen, it's a proton. It's part of a hydrogen. And the electrons are designated by the E's, electrons. So they have a negative charge, the protons have a positive charge. So what happens in the electron transport system is the hydrogens are delivered, split into its atomic components, electrons and protons, and then the electrons are shuttled along from one enzyme to another in the membrane of the mitochondria. And then the protons, they follow a different pathway. And as a result of the combination of what these electrons are doing and what these protons are doing, then ATP can be generated. A lot of ATP. So that's what we're going to see. We're going to look at the electrons and what they're doing, and then we're going to look at the protons and what they're doing. So first of all, these electrons, remember, they're split from the hydrogen. You've got these electrons. Electrons have energy. They have different energy states. And so as these electrons pass from one enzyme to another, if you look in this figure, there's these big white things that are in the membrane are enzymes. And the magenta pinkish colored lines show what happens with the electron. So an electron is passed from one enzyme to another 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 enzyme. And if you look over there on the right in this figure that shows the amount of energy, the free energy that's in electrons, then what happens when the electrons are passed between these different enzymes, they give up some energy. They lose energy. So the units of energy in that figure are high when this whole series of electrons being shuttled starts, and they're a lot lower when this whole process is done. The electrons are being shuttled along, transported along between different enzymes, and they're giving up energy each step of the way. That's where you see these H pluses. The energy that's being given up by the electrons is being used to do something with the protons. So that's what we're going to look at. In fact, this energy that's given up when you drop down energy state, and an energy state is used to pump the protons against their concentration gradient. Remember, moving substances against a concentration gradient, like carrying something up the stairs, takes work. It takes energy. It's not ATP that's the form of energy in this case. It's the energy that's given up by these electrons. So the electrons are giving up a little bit of energy each step of the way. That energy is used to pump protons against their concentration gradient across the mitochondrial membrane, and these protons build up in concentration in the space between the two mitochondrial membranes. So if you look in this figure, each one of these little H pluses, the gray lines, are being pumped through these enzymes, and they're showing up on the outside of this inner mitochondrial membrane. So you have 2H plus, 2H plus, 2H plus, 6H plus over here. These are building up as they're being transported. Here's another way of looking at this. This is what's happening with the protons. The electrons are giving up some energy. They're losing energy as they're being shuttled between these different enzymes. Now that energy that's being liberated by the electrons, it's shown here in these big black arrows that are protons being transported against a concentration gradient, uphill, not down with no energy required, but up with the energy requirement. And so look at all these H pluses that are up here on this blue part of this figure. There's a lot of them there, and there's not very many on this red part. So now you have a gradient of protons, which means you've got a high concentration of protons on one side of the mitochondrial membrane. You've got a low concentration on the other side. This concentration gradient is attained because of the energy that electrons have given up. And remember, both of these components, protons and electrons, have come from the hydrogens that we ate in sugar. So now the energy has gone from glucose to pyruvate to acetyl-CoA, and some to ATP directly, but a bunch of it has been stored temporarily in those hydrogens. Now those hydrogens are giving up their energy in the form of electrons, first of all, that are giving up some energy that's transferred to protons. It's not stored in the protons themselves, but that potential energy is stored in the concentration of protons, all these protons that are in that blue area in this figure. There's some potential energy there. There's a high concentration here and a low concentration there. We'll see in a minute how this is used. Now, here's another way of looking at this. Again, concentrating on the electrons and the protons. So these electrons are being shuttled along, giving up some energy from one enzyme to another enzyme to another enzyme, another enzyme, another enzyme, another enzyme, another enzyme. Each step of the way they're giving up energy. That energy is used to pump these protons. Well, when the electrons are done being shuttled between the enzymes, you've got a whole bunch of electrons. What happens with these? We can't take in an endless supply of electrons. You've also got these protons that are doing this, being shuttled across the mitochondrial membrane, and we'll see in a minute how those protons are used. But at the end of this process, you've got electrons and you've got protons. Now, where did those come from? They came from hydrogen. So if you combine those again, an electron and a proton, then the product is a hydrogen. So you have all these hydrogens that we've taken in as food. We saw how we got rid of the carbon and the oxygen in glucose, but not the hydrogen. This is what happens. The electron and the proton join together to form a hydrogen, and that hydrogen forms a bond with oxygen. 2 hydrogens for each oxygen molecule. There's oxygen that we breathe in that's distributed to our mitochondria that keeps this process going. This oxygen is the final electron acceptor. So you have these electrons combine with oxygen, and then you have the protons combine with the oxygen. So what you form is H2O. 2 hydrogens, 1 oxygen. It's water. So first of all, the oxygen is used to help get rid of the electrons and get rid of the protons. It's a mechanism for getting rid of all that excess hydrogen that we've eaten. And also, oxygen was building up in the atmosphere as photosynthetic organisms developed billions of years ago. And oxygen was quite toxic. It was a bad thing, this oxygen. So this is another mechanism for organisms dealing with that somewhat toxic gas of oxygen. They can use oxygen, combine it with hydrogen, and what are they making? The most innocuous thing that you can, water. Water is what you make. And so I want to point out 2 things here. One is that the fate of oxygen that we breathe in is not carbon dioxide. We breathe out carbon dioxide. We saw earlier in the Krebs cycle that carbon dioxide comes from sugar, not from breathing in oxygen. This oxygen leaves our body not as carbon dioxide like most people think. It leaves our body as water. So it encompasses a number of different things, but the end product is water, and we make water. We can use that water. We can get rid of that water. It's a good byproduct of cellular respiration. That one of the waste products is water, because there's not much of a waste product at all to us. Okay, so we saw what happens with these electrons. Now let's look at what happens to these protons. We saw these protons are pumped across the mitochondrial membrane. They're pumped across against their concentration gradient. And so that's what we see here, is these protons being pumped up here, and now this time it's all these H pluses that are building up here. There's a lot of H pluses up here, not very many here. And these big arrows that are coming through these enzymes represent protons being pumped against their concentration gradient with energy that's come from the electrons. And now what these protons do, they can't get through this membrane. This membrane is very impermeable, even to tiny little protons. So there's a high concentration here. They would go through this membrane down their concentration gradient if they could, but they can't. There's one way that they can get through this membrane, and that is this enzyme that has a channel that allows the protons to go through and go down their concentration gradient. That enzyme is called ATP synthase because it makes ATP. It's an enzyme that synthesizes ATP. It's not making brand new ATP. We looked at this a while ago. It's taking a phosphate and joining that to ADP to make ATP. You can see that if you look closely here. The energy that's liberated when these protons pass down the concentration gradient, that represents energy. That energy is used to take that phosphate and stick it on an ADP and make ATP. And there's a lot of energy that flows through these ATP synthase enzymes. That is how you make so much ATP in the electron transport system, because there's all these hydrogens that are broken apart to produce all these electrons that are used to pump all these protons against their concentration gradient. And all these protons then pass through this enzyme ATP synthase, and they make a lot of ATP. Well, one analogy to this is a water behind a dam. You guys are all familiar with this. There's these big giant dams, especially out west, and there's big giant reservoirs behind these dams. They let the water spill through these dams and turn the turbines, and they make electricity. That's how you get a hydroelectric power. Well, it's a similar situation here. If you look at this, this is Hoover Dam over there on the border of Arizona and Nevada, I think. All these protons up here in the blue represent a surplus of protons, a high concentration. It's like water behind a dam. The dam is analogous to the cell membrane or the mitochondrial membrane because those protons can't get through. The spillway, where the water goes through and turns the turbines, is like this ATP synthase. Instead of water flowing through, it's protons. Those protons turn this turbine and carry out this reaction, or they power this reaction, ADP plus inorganic phosphate, to make ATP. So in that way, this whole mechanism, this whole setup of high concentration on one side and low concentration on the other side, and some kind of a passageway, is similar to how we make electricity at these hydroelectric dams.